But with that, I'll go ahead and move into the first method that I'm going to talk about today, which is the popular method gene set enrichment analysis, or GSEA. So GSEA is originally an online tool and also a desktop software application that you can access from this link on the page. But very generally, it determines if a ranked list of genes is significantly associated with some biological function represented by a gene set. So you'll notice that GSEA does require a ranked or ordered list of genes or a ranked gene signature as the input. This is a brief overview of the steps. I'll go into a little bit more depth about some of these steps later on, so you don't need to, try to write all this down right now. But essentially, you can kind of look at the figure on the right, and you'll see that you have some sort of ranked gene list that you've obtained from, maybe a study that looks at two different phenotype classes, A and B. And then you want to compare the genes that you've ranked in that signature with some comparison or annotated gene set S. And essentially, you'll compute a running sum or a Brownian bridge or random walk plot. And you'll do that by basically placing your sorted ranked input genes along the x-axis of that plot on the very far right. And then you compute the plot, and the peak of that plot, will actually be your enrichment score. And then you can determine the significance of the relationship between the ranked list and your annotated gene set afterwards. So we can start with a relatively simpler example. So let's say that we have samples from two classes, A and B, that we're trying to compare. So then you generate a signature where the genes are ranked by their differential expression. And that was what was covered last lecture. So use any of those methods and you generate the signature. And now you want to compare your signature to a gene set, which we'll just say for simplicity's sake is just the genes B and C in this set. After you rank the genes, the next step would be to generate this random walk plot to compute the enrichment score. And essentially, this random walk plot that is used in GSEA is built upon this idea of Brownian motion, which is the random motion of particles at thermal equilibrium. And the original traditional example is that you start at 0 on a one-dimensional number line, and then you're randomly taking steps left or right. And then you can plot the displacement from 0 as time goes on, and you'll see that generally the trend is towards increasing displacement. The Brownian bridge plot then, which is what our random walk plot is based on, is built on that same concept of Brownian motion, except that the start and the end points of the walk are fixed at 0. So you'll start and end at 0, but in the middle, the displacement will continue to be sort of random. And essentially the supremum of this plot would be the maximum displacement of this walk over the Brownian bridge. So wherever you have the greatest displacement from that 0 point. If you're interested in the mathematical specifics, definitely read the original GSEA paper or read the paper that I cited on this page from where I took this actual example that I'm showing. But in general, the distribution of the displacement in a random walk follows a normal distribution. So you can kind of think of it as generally, if you're bound at 0 at both ends, your displacement will tend to be somewhere in the middle usually. You're less likely to be very, very close to 0 or very, very far from 0. And then you can look at the cumulative distribution functions, which basically shows for each displacement value, what is the probability that the displacement of the point at a given time is less than that displacement value from 0 in a random walk plot. And because we expect that the distribution should follow a normal or a Gaussian distribution, we can look at the difference between the cumulative distribution function of a normal distribution, which is what we expect from a random walk, a truly random walk. And then we can take the difference of that and the cumulative distribution function we actually observe from our data. And this is essentially just what the Kolmogorov-Smirnov test evaluates. So here I can kind of show an example what it would look like if the random walk is not actually random. So previously you saw that the cumulative distribution function of the data on this slide pretty much matches with the hypothesized normal distribution CDF. But here, if the data is not actually consistent with a normal or Gaussian CDF, so your data is not actually your displacement is not actually following that normal distribution, you will get a biased plot in terms of your CDF and also in terms of the random walk plot. So you can see that there seems to be a kind of an arc above zero here at the bottom. So how you actually generate the random walk plot in practice, we can show that using the example that I set up a few slides ago. So first we can quote unquote walk down the list of ranked genes with the first gene placed at, x equals 1. And essentially, if a gene is not in the comparison gene set, which again, in this case, is just the set B and C for simplicity, then we want to decrement the value of the plot. So here I'm just decrementing by negative square root of G over N minus G, where G is just the number of genes in comparison set, and N is the number of genes in our data. So you'll see that the first 3 genes in our ranked list are not in the comparison gene set, so we're decrementing this plot. And then we want to increment the plot if the gene at a given position is in the comparison set. So here you see that C and B are both in our comparison set. So we end up actually incrementing the plot. And then the supremum of that random walk plot is the enrichment score. And you'll see here that we actually technically have a negative supremum, which is why we don't say maximum or minimum, because it could be positive or negative. And how to interpret the enrichment score is as follows. So if you have a positive supremum or a maximum score, then that indicates the genes in your comparison set tend to be upregulated in the data. And note that when your ranked list of genes is from GWAS or ChIP-seq studies, you should only ever consider it a positive supremum because you only have positive hits. If you have a negative supremum or enrichment score, like we saw in our example, that indicates that the genes in the comparison set tend to be downregulated in your data. And then if you have like no significant deviation from y equals 0, and I'll talk more about significance later, that indicates that the genes in the comparison set do not appear to be significantly differentially expressed in your data. So just for some practical examples from the original GSEA paper. So this plot basically shows the random walk plot for a signature between male and female lymphoblastoid cell lines, which is compared against a set of chromosome X inactivation genes. This is a sex-based trait, so you'll see that these genes are significantly enriched in the female samples based on the plot. And then you can see that across the top, there's this horizontal line with the vertical dashes. So each of those dashes actually indicates like a gene hit, or basically where genes in the comparison set were ranked in that signature or ranked list. And you'll see that they're very highly concentrated toward the beginning of the plot. So they're very upregulated in this signature. For a pathway that is not really differentially expressed between male and female cell lines in this case, we're looking at vitamin C import pathway genes. So note that even though this was a comparison between male and female samples, it is possible that this pathway could have been, differentially expressed, but here we see that it's not, and you can tell from both the plot itself of the random walk, you can see how it's not really deviating from 0 in the same way that the previous plot was. And also by looking at that plot of the gene hits across the top, where you have the horizontal line, the vertical dashes, the genes in the comparison set are pretty evenly spread across the rankings from your signature. So there's not really any significant enrichment here. But I mentioned significance a couple of times. I want to make sure I go over how significance is actually estimated. So I think essentially the traditional way of doing this is to randomly swap the class labels of your samples and then repeat the process I just talked about many, many times, usually probably like 1,000 different permutations. And then the significance can be calculated from the number of times when your observed enrichment score is larger than the enrichment score you got from these random permutations, if that makes sense. So in summary, GSEA is a very useful tool and a very popular tool for computing enrichment analysis for a ranked gene list that's usually derived from a comparison between two conditions. The GSEA algorithm produces an enrichment score that's based on the supremum of a Brownian bridge or random walk plot. Significant results can be quantified by comparing that given enrichment score against repeated random permutations of class labels and the enrichment scores obtained from those permutations. And in general, you won't really ever need to know like every specific step or the very minute mathematical details of this process because there's a lot of different implementations online. So it is originally a web tool that you can use or a desktop tool. There are also R implementations. And then there are two Python implementations, GSEA Py and then Blitz GSEA, which was developed by our lab, which are also available. And I'll go over those in the demo later.